Good morning, students. This is your teacher Neelam Helawat, and we'll continue with the chapter. This is our lecture number 4, and we'll start with the search of genetic material. This is our topic for today. So you keep your notebooks ready. We'll start with the topic. Now see, we know that it was Gregor John Mendel who experimented with the pea, and he talked about that there are certain factors which are being transmitted. And these factors they constitute the gene, and genes are present on the chromosomes, right? And during cell divisions, 50% of the genetic material is being transported. During gamete formation, only 50% of the genetic material is packed into the gametes. And the biochemical studies have revealed that the chromosomes, they are made up of proteins and DNA. This information we know today, but initially there was no clarity was there that what is the genetic material. This search had took a long time to establish that DNA is a genetic material. So we'll talk about those experiments that led to the establishment of the genetic material. Okay. So initially the genetic material should be able to it should be able to replicate itself. Replicate means it should be able to produce a copy of itself. Second, it should be able to store the information that is needed for the transmission of a particular character. It should be able to control the traits, and it should be able to undergo the changes that may be due to the mutations or may be due to the environment, and it should be a stable one. So anything which act as a genetic material should have these 5 characteristics. Okay. Now, is the genetic material a protein or the DNA? This was the concept which was going on, and which was debatable at that point of the time. Some of them, they considered that the protein was the genetic material. Some of them, they argued that the DNA is a genetic material. And the third category also argued that not the protein, not the DNA, but the RNA is the genetic material. Okay. So until 1940s, the protein was considered to be the genetic material. As a proteins, there are 20 amino acids. Okay. Proteins are the polymers of 20 amino acids, and they encode a variety of information. DNA is a polymer of 4 different deoxyribonucleotides. This we have talked about. There are 4 bases we have seen so far, that adenine is there, threonine is there, then guanine, then a cytosine is there, right? So DNA is polymer of 4 different deoxyribonucleotides, and most geneticists, they focused on transmission of genetics, and they passively accepted proteins as a genetic material. So without any debate, they had accepted that the protein is the genetic material. But on the basis of the certain experiments that were conducted time to time, it was evident that DNA is the genetic material. Right? These evidences were, some of them, they were the direct evidence, some of them, they were the indirect evidence. The 3 direct evidences are basically the 3 experiments. One is first as the Frederick Griffith's experiment. Second was Avery, MacLeod and McCarty's experiment, and the third is considered to be the very, very important that led to the establishment of the DNA as a genetic material is Hershey and Chase experiment. These 3 experiments together establish the fact that DNA is a genetic material. Now, very first is Frederick Griffith experiment. Now, Frederick performed his experiments on Diplococcus pneumonia. This was the organism, or you can say this was the experimental organism on which the Frederick performed his experiments. Now, this Diplococcus pneumonia has 2 strains. One is the R strain, another is S strain. Okay, so this is simply your S strain. Okay, this is your S strain. And this S strain is virulent. Virulent means it causes a disease. Okay. Now this S strain it has a liposaccharide capsule. जो हमारा S स्ट्रेन है ये एक तरीके से इसके ऊपर इसके लिपोपॉलीसैकेराइड का कैप्सूल होता है and it causes the disease, and because of the disease the organism can die. Okay, so this causes a disease which is called pneumonia. ठीक है, तो हमारा डिप्लोकोकस निमोनिया जो है इसकी दो स्ट्रेन्स हैं, वन इज़ एस स्ट्रेन, अनदर इज़ आर स्ट्रेन. जो एस स्ट्रेन है इससे बीमारी कॉज होती है, ये बीमारी को कॉज करता है, इसीलिए हम इसको वायरुलेंट कहते हैं. इसके ऊपर एक लिपोपॉलीसैकेराइड का कैप्सूल होता है जिससे इसका सरफेस स्मूथ नज़र आता है और इसकी वजह से बीमारी होती है निमोनिया. द सेकेंड स्ट्रेन इज कॉल्ड द आर स्ट्रेन. ओके, इट डज नॉट हैव अ पॉलीसैकेराइड कैप्सूल. इसके ऊपर जो है पॉलीसैकेराइड की कोटिंग नहीं होती है, इसलिए इसका सरफेस जो है वो रफ होता है. दैट इज़ वाई इट इज़ कॉल्ड अ रफ स्ट्रेन. ओके, सो इट डज नॉट हैव अ कोटिंग एंड दैट कोटिंग इज लिपोपॉलीसैकेराइड कोटिंग एंड इट इज़ अ वायरुलेंट. मतलब ये वो स्ट्रेन है जिससे बीमारी कॉज नहीं होती. Okay, so we have R strain, we have S strain. R strain means your rough strain. Okay, here the capsule is absent and it does not cause any disease. Second is S strain, means it is smooth, the surface is smooth, it contains a capsule and it causes the disease. So we can compare the two strains very well. R is not causing the disease while S is causing the disease. Now this was his experiment. What he basically did, in first part what he did, he induced the S strain into the mice. ठीक है, तो सबसे पहले उन्होंने क्या किया, उन्होंने माइस के अंदर S strain को इंजेक्ट किया. When S strain was injected, the mice died because the mice has developed the disease pneumonia and the mice died. In second stage what he did, he induced the R strain into the mice. पहले उन्होंने कौन सा स्ट्रेन इंड्यूस किया था, S strain. सेकेंड टाइम उन्होंने R strain को माइस के अंदर इंड्यूस किया and there was no disease, so mice was alive, it was living. Third time what he did, in third time he mixed the two strains. उन्होंने S और R strain को मिक्स किया. ठीक है, उन्होंने क्या किया कि जो S strain है, इज इट क्लियर, जो S strain है उसको हीट किया. थर्ड स्टेप में उन्होंने S strain को हीट किया और हीट किल्ड S strain को उन्होंने इंजेक्ट किया. ठीक है, फर्स्ट उन्होंने S strain को इंड्यूस किया, सेकेंड उन्होंने R strain को इंड्यूस किया, थर्ड उन्होंने अब क्या किया, S strain को हीट किया. हीट की वजह से जो S strain है दे बिकम डेड. जो डिप्लोकोकस निमोनिया था इट डाइड and then it was injected into the mice and the mice was living. Clear? This means जब हमने S strain को हीट किया तो S strain में जितने भी निमोनिया थे they died. The Diplococcus pneumonia, this organism, they died and the mouse was alive. Then the last stage what he did, he mixed the R strain plus the heat killed S strain. ठीक है, उन्होंने फोर्थ में क्या किया, जो S strain था जिसको उन्होंने हीट किया था, जो डेड था, हीट किल्ड S strain प्लस R strain को मिक्स कर दिया. ठीक है, तो इन्होंने S और R strain को, S कौन सी स्ट्रेन जिसको हीट किया था, इन दोनों को ऐड कर दिया, मिक्स कर दिया एंड देन ही इंजेक्टेड इनटू द माइस. ठीक है, इज इट क्लियर? S strain वो है जिससे बीमारी होती है, R strain वो है जिससे बीमारी नहीं होती. तो जिस पर्टिकुलर स्ट्रेन से बीमारी होती है वो हीट किल्ड है. ओके, एंड आर इज प्रेजेंट, सो आर से बीमारी नहीं होती. बट ही वॉज सरप्राइज, इट वॉज द रिजल्ट वॉज सरप्राइजिंग बिकॉज द माइस डाइड. ठीक है, अगर हीट किल्ड इन थर्ड केस ही हैज़ इंड्यूस्ड ओनली द S strain, heat kill S strain into the mice, but the mice was living. Okay, and in fourth case he had mixed heat killed smooth bacteria with rough ones, but now this time the mice was dead. Okay, the mice was dead. let's again see. First, he injected the S strain, the mice was dead. Second, he injected the R strain, the mice was living. Third, mouse was injected with heat-killed strain, the mouse survived. Then he mixed the heat-killed S strain with R strain, the mouse was dead. And there was observed that in the blood the S strain was present. और ये भी ऑब्जर्व किया गया साथ में कि जो माइस का ब्लड है उसमें एस स्ट्रेन मौजूद है. सो हाउ कुड इट बी पॉसिबल? देन Frederick Griffith, he concluded that the R strain bacteria had somehow been transformed by heat killed bacteria. Means जो S बैक्टीरिया हैं जिनको हमने हीट किया था, जो डेड थे, उनमें कोई ऐसा फैक्टर था, बिकॉज ऑफ दैट फैक्टर द R strain has been converted into or transformed into S strain. ठीक है, R strain डिजीज़ कॉज नहीं कर सकते हैं. S strain जो है वो ऑलरेडी डेड थे, तो S strains में कोई ऐसा फैक्टर मौजूद था जिनकी वजह से R strain जो है वो S में ट्रांसफॉर्म हो गए. And these transformed S strain, right? These transformed S strain became virulent, and that is why the mice was dead. Thus, this must be the genetic material. तो वो फैक्टर जो S हीट किल्ड S strain में मौजूद था, जिसने R strain को S में कन्वर्ट कर दिया है, वो फैक्टर ही जेनेटिक मटीरियल है. ओके, ओनली दैट फैक्टर इज द जेनेटिक मटीरियल. हाउएवर, इट्स बायोकेमिकल नेचर वाज नॉट डिफाइंड. ठीक है, बट दे कुड नॉट डिफाइन इट्स बायोकेमिकल नेचर. Then came the second experiment, that is by Avery, MacLeod and McCarty. Now this was also based on some transformation. They obtained some free extract from the S strain bacterium. अब इन्होंने क्या किया, जो सेल कल्चर था उसमें से S strain bacterium को obtain किया, and they subjected to DNA. They subjected, these are 3 enzymes, DNase, RNase and protease. ठीक है, DNase which will able to catalyze the DNA, RNA the enzymes which will work upon RNA, and protease which will work upon the proteins. ठीक है, and they each extract, जो DNA जो एक्सट्रैक्ट ऑब्टेन किया, RNA से जो ऑब्टेन एक्सट्रैक्ट बनाया, प्रोटीज से जो एक्सट्रैक्ट बनाया, ईच एक्सट्रैक्ट वर देन इंजेक्टेड इनटू द माइस. ओके, सेपरेट माइस में सैम्पल्स को इंड्यूस किया गया. ओके, इन केस ऑफ प्रोटीएज एंड RNase, ट्रांसफॉर्मेशन वॉज रिकॉर्डेड. So in case of protease, that is the enzyme protease and RNase, the transformation was recorded. But in case of DNase, no transformation was recorded. Means जिन enzymes ने DNA को break down किया था वहाँ transformation नहीं हुई. Clear? So this again they concluded from the experiment that the removal of all proteins from the transforming material did not destroy its ability. कि अगर हम proteins को destroy कर देते हैं, ठीक है, अगर हम प्रोटीन्स को डिस्ट्रॉय कर देते हैं, प्रोटीन्स को डिस्ट्रॉय कौन सा एंजाइम करता है? प्रोटीनेज. ठीक है, अगर प्रोटीनेज जो है सारे प्रोटीन्स को डिस्ट्रॉय कर देता है तो R स्ट्रेन्स की एबिलिटी, ट्रांसफॉर्म होने की एबिलिटी डिस्ट्रॉय नहीं होती है. ठीक है. सेकेंडली, DNA डाइजेस्टिंग एंजाइम्स जब DNA को ख़त्म कर देते हैं, डाइजेस्ट कर देते हैं, then the ability to transform is destroyed completely. Okay, then from this experiment it was concluded that the transforming material is DNA. But the full and the final experiment that established the fact that DNA is a genetic material was given by Hersey and Chase experiment. This was very, very important. Now what these two scientists they did, they conducted their experiment on bacteriophages. Bacteriophages क्या होते हैं? ये वो वायरस हैं जो बैक्टीरिया को इन्फेक्ट करते हैं. क्लियर? These are the viruses which infect the bacteria. That is why the name bacteriophages. ठीक है? And the bacteriophages, they are made up of DNA and proteins. ये दोनों चीज़ों से बने होते हैं. DNA and proteins दोनों होते हैं इनमें. Now they wanted to determine that out of DNA and protein which is actually the genetic material. Okay? So DNA और protein दोनों में से कौन सा exactly genetic material है. So they conducted this experiment. Now we must understand the life cycle of bacteriophage, that is T2 phase. Now this is made up of DNA and proteins, okay, and we know that if DNA is a genetic material, then only DNA will enter into the nucleus. Clear? And if RNA is the genetic material, if protein is the genetic material, only protein will enter. So they wanted to see it. Clear? Generally, life cycle में क्या होता है? So this portion you see, this is your bacteriophage and this is the bacterial cell. So bacteriophage will infect the bacteria. It will attach on the wall of the bacteria. Okay? And it will inject its genetic material into the bacterium. ठीक है? बैक्टीरियोफेज जो है वो बैक्टीरिया के सेल सरफेस पे अटैच होगा और अपना जेनेटिक मटेरियल वो सेल के अंदर इंड्यूस करेगा. दिस बैक्टीरियल सेल विल देन मेक मल्टीपल कॉपीज ऑफ द जेनेटिक मटेरियल विच इज प्रेजेंट इन of bacteriophage, right? Clear? And this bacteriophage will be then released from the surface. They will multiply inside and then these bacteriophages, they will be released. Okay, this is the general life cycle. Clear है सबको? बैक्टीरियोफेज जो है वो बैक्टीरिया के सर्फेस पे अटैच करेगा, अपना जेनेटिक मटेरियल बैक्टीरियल सेल के अंदर इंड्यूस करेगा. बैक्टीरियल सेल इस जेनेटिक मटेरियल को डुप्लीकेट करेंगे, मीन्स दे विल इंक्रीज द नंबर ऑफ bacteriophages and these bacteriophages, they will be released. Clear? Now अब हमने क्या किया? They both did. The bacteriophage was grown in 2 mediums. ठीक है, हमने 2 bacteriophages को 2 मीडियम में ग्रो किया. In one we have made the phosphorus radioactive. ठीक है, अब हमने 2 मीडियम में bacteriophages को ग्रो किया. In first medium we have phosphorus radioactive. Phosphorus was radioactive. ठीक है, हमने phosphorus 32 को रेडियोएक्टिव जो है उसको इंड्यूस किया जिससे कि हम डीएनए को रेडियोएक्टिव बना सकें. नाउ व्हाई वी हैव टेकन phosphorus 32? हमने डीएनए का स्ट्रक्चर डिस्कस किया है एंड वी नो दैट डीएनए इज मेड अप ऑफ 3 थिंग्स, राइट? क्या क्या होते हैं वो? शुगर होती है, नाइट्रोजन होता है, नाइट्रोजन बेस होता है, and what else? Phosphate group. Okay, so we have understood. So DNA includes the phosphorus. Okay, so in one medium we made the DNA radioactive by inducing phosphorus 32. In second medium we made the proteins radioactive by inducing sulfur radioactive, sulfur 35. तो हमने बैक्टीरियोफेजेस को 2 मीडियम में ग्रो किया. एक मीडियम जिसमें हमने डीएनए को रेडियोएक्टिव बनाया बाय यूजिंग द फॉस्फरस 32 और एक मीडियम जिसमें हमने प्रोटीन्स को रेडियोएक्टिव बनाया बाय टेकिंग द सल्फर 35. ओके, क्लियर? नाउ दीज bacteriophages from both the mediums were taken and these were allowed to infect the bacteria. अब ये दोनों मीडियम में से हमने बैक्टीरियोफेजेस लिए और हमने बैक्टीरिया को इन्फेक्ट किया. ठीक है, इज इट क्लियर? लेट्स सी दिस. सो हियर इन दिस फर्स्ट केस वी आर टेकिंग phosphorus 32, यानी हमने डीएनए जो रेडियोएक्टिव किया है वो, एंड हियर वी आर टेकिंग sulfur. Okay, sulfur 35. In first case we have made the proteins radioactive and here we have made DNA radioactive by taking phosphorus 32. Let's see the first one. Now this is a bacteriophage. This is bacteria. Bacteriophage will attach with the bacteria, will induce the material into it and separate out. So देखिए material induce हो गया, ठीक है. And यहाँ पर अब हम will analyze the DNA or the genetic material present in the bacterial cell. हम bacterial cell के अंदर analyze करेंगे, ठीक है. When we analyzed this bacterial cell, there was no radioactivity was observed. ठीक है, no radioactivity was observed. I'm again repeating. यहाँ पर हमने प्रोटीन्स को रेडियोएक्टिव बनाया था because by inducing sulfur 32. So bacteriophage, it will attach with the surface. It will put the genetic material into the bacterial cell. Now we'll analyze the bacterial cell to observe the radioactivity. But the genetic material did not have any radioactivity. यहाँ पर हमने ऑब्जर्व किया कि जो जेनेटिक मटीरियल है उसके अंदर रेडियो एक्टिविटी नहीं थी. क्लियर? In second case, where we have made the DNA radioactive by taking phosphor 32. Now these were also allowed to infest the bacterial cell. Okay. So there was first test was infection, then there was blending. Blending means the two genetic material, that is the genetic material of bacteriophage plus the genetic material of bacteria. They will blend with each other. They will mix with each other. Right? Clear? And now we centrifuged it to observe the radioactivity in the DNA, in the genetic material. Okay. In this case, highly radioactive cells were detected with radioactive phosphorus 32. Okay, the cells were detected with the bacterial cells. They were detected or they contained most of the radioactive P32. So from this experiment, it was concluded that it is the DNA which is the genetic material. It is not the proteins which are the genetic material. So the results of this experiment clearly indicate that only DNA, nor the proteins, they have entered the bacterial cell. कि जब बैक्टीरियोफेजेस ने बैक्टीरियल सेल को इन्फेक्ट किया है तो उन्होंने अपना डीएनए मटीरियल, अपना डीएनए बैक्टीरियल सेल के अंदर इंड्यूस किया है, एंड दैट हैज़ मल्टीप्लाइड. The DNA on entering the host cells carry the genetic information for the synthesis of new phage particles. Does this prove that only DNA is the genetic material in bacteriophages and not the proteins? So this experiment was highly, or you can say it is very crucial experiment that established the fact that only DNA is the genetic material. No other substance has the ability to act as a genetic material. Clear, students? Okay. So this will be your homework for today. First of all, you will make notes of the topic. Then you will read the page numbers 100 to 102. These are all three experiments that we have discussed. You will all underline each and every line. You will note down the important points in your books. Okay? And please do practice the questions which are given in the Meritination. Okay? Extra marks. These are the two sites where extensive work is given. You can go through their study material also if you want. Okay? Bye everyone.